This video is going to cover the endomembrane system. So first let's take a look at this word endomembrane. So endo means inside or within. And the membranes that we're talking about here, we're talking about the membrane-bound organelles that are found in eukaryotic cells. So as I mentioned, the endomembrane system is primarily associated with eukaryotic cells. And that's because eukaryotic cells have these membrane-bound organelles, whereas prokaryotic cells do not. There are several organelles that are associated or incorporated in this endomembrane system. The first one is the nuclear envelope or membrane. And then we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And we can abbreviate those as the RER and the SER. We also have the Golgi apparatus. And then in animal cells, we also have lysosomes. We also have transport vesicles. And then I'm going to go ahead and add the cell membrane kind of in brackets here because the cell membrane is not technically inside the cell, it's around the cell. So it's not technically part of the endomembrane system, but all of these membrane-bound organelles and especially the membrane components of them interact with the cell membrane. So it's important to talk about it as we talk about the endomembrane system. Now, we are going to go over a little bit of information about the structure and function of these different organelles here. But if you haven't learned about the structure and function of them already, I recommend that you pause the video and go look up the structure and the function of these organelles. So look up the structure. So the structure is what it's made out of, what it looks like. And also look up the function, which is what it does. It is also important to note that the endomembrane system does not include all of the membrane-bound organelles. For example, mitochondria and chloroplasts are not part of this system. So even though the mitochondria and chloroplast are membrane-bound organelles, they are not part of the endomembrane system. So when we talk about these membrane-bound organelles and the cell membrane, we can kind of generally refer to the membrane or the boundary as the plasma membrane. And we will talk more about the structure of the plasma membrane when we talk more about the cell membrane. So for the sake of this video, we're just going to draw this plasma membrane as a line or a circle. But it's important to note that this plasma membrane is very fluid. And what I mean by this is you can kind of think of it as bubbles. So if you've ever played with bubbles as a kid, you know that you can take multiple bubbles and you can kind of combine them together to form a bigger, like a single bigger bubble. You can also take part of a bubble and kind of pinch it off into a smaller bubble. Granted, the soap bubbles that you played with as a kid were pretty delicate and would pop pretty easily, and that's not the case with the plasma membrane. Instead, we have these kind of fluid membranes that can fuse with each other, but also pinch off from each other. So I mentioned for simplicity's sake, we're just going to draw this plasma membrane or this membrane as kind of a line or a circle. So if we were to look at a structure here, it doesn't matter what the structure is, but the line is going to be our membrane. Now let's say we had another structure that also had a plasma membrane. What could happen is that these two structures could actually combine and kind of fuse together until they ultimately form just one big structure. On the flip side, the opposite could happen. So you could have a structure that has a little part of the cell membrane that actually starts to kind of pop out from it. So we could kind of draw that as something kind of like this. And so this area right here is where that membrane is starting to pinch off. And ultimately what can happen is we can end up with the original structure plus this little piece that has popped off like a little bubble. So keep this in mind that part of the membrane from one structure might be able to kind of pock out and then pinch off, and then it can be transported to another structure or another organelle that has that similar membrane, and it can fuse with that in order to basically add itself into that new membrane. So let's use some colors to kind of go over this concept again to maybe help explain it better. So let's say that one of these membranes, and I'm going to kind of highlight it in yellow, and then the other structure with its membrane, I'm going to highlight in green. So as they fuse together, what we end up with is that yellow membrane combining with that green membrane. And so our final structure is going to contain components of each of the original membranes. Now note that if there is material inside these different structures, that material is now going to be exposed to each other. And so we end up with this kind of mixture of the two structures that have combined together. On the other example, we could take something like this. So now this structure, we've highlighted the membrane in yellow. And then as the other structure kind of pinches off from it, it's still going to be containing that same membrane, so that it's just the same membrane that is pinching off. Once again, if there's material inside this structure, some of that material might end up in the final little piece that pinched off there. All right, so let's draw out this endomembrane system in kind of a cartoony way. So first of all, we're just going to draw a big circle, which represents the cell membrane. Inside a eukaryotic cell, we have a nucleus. And that nucleus actually has a double membrane. So we'll draw two lines like that. Now these two membranes are continuous with each other. So they're two layers, but it's kind of the same membrane kind of folded in on itself. So this would be the nucleus. And this area here would be the nuclear membrane. And it's actually two layers of membrane. Now, continuous with the membrane of the nucleus, we're going to see another membrane-bound organelle called the rough endoplasmic reticulum. So kind of coming off of that nucleus, we have this structure called the rough endoplasmic reticulum. And it's called rough because it has these ribosomes stuck to the cytoplasmic side of the membrane. And then continuous with the rough ER is actually the smooth ER. And so the smooth ER has a similar kind of structure to it. The main difference is that it does not have those ribosomes. The next structure associated with the endomembrane system is going to be the Golgi apparatus. And the Golgi apparatus looks like a stack of pancakes, kind of. So it is made up of these kind of multiple skinny saccules that are stacked on top of each other like pancakes. And so this whole area or structure is called the Golgi apparatus. So now that we've established kind of what these structures look like, let's talk a little bit more about how they are related to each other. So first of all, if we look at that rough endoplasmic reticulum, we know that ribosomes make proteins. And so the rough endoplasmic reticulum, it kind of serves as a protein factory, and those proteins are either going to be transported outside of the cell or they're going to become part of the cell membrane. So what happens is that these ribosomes make proteins, and these proteins get kind of delivered to the inside of the rough endoplasmic reticulum. So they actually end up either inside this membrane-bound organelle, or sometimes the proteins get embedded in the membrane of the rough endoplasmic reticulum. So what happens is that part of the membrane from the rough endoplasmic reticulum can actually pinch off into a transport vesicle. So now this vesicle either contains a protein inside of it or a protein embedded in the membrane of it. Now, the membrane of this vesicle, remember these membranes are very fluid and they can combine with each other and they can separate from each other. So the membrane of this transport vesicle can actually go to the first part of the Golgi apparatus. And the edge of the Golgi apparatus that is on the same side as the rough endoplasmic reticulum is called the cis side. And then the edge or the side furthest away from the rough endoplasmic reticulum is called the trans side. So what happens is that this transport vesicle ultimately ends up fusing with the membrane of the Golgi apparatus. And so the protein is now inside the Golgi apparatus, or it's embedded in the membrane of the Golgi apparatus. Now, the Golgi apparatus has a couple of different functions. So one function is that it modifies the different products that it receives. It also packages them, and so it sends them to their final destination. So we typically think of the Golgi apparatus as kind of acting like the post office. So what we mean by modifying is that it might add a little piece or something to that membrane. It might cause it to fold in a certain way, et cetera, et cetera. So modify usually means that it's adding a molecule or a molecular component to that protein. For example, it might be adding something like a carbohydrate. And this helps to form things called glycoproteins. It also helps with folding the protein into its final three-dimensional shape. Now, transport vesicles can then move these materials through the different stacks of the Golgi apparatus. So we can draw another transport vesicle transporting that modified protein into the next stack. And then this continues kind of all the way down. So ultimately, we would end up with another transport vesicle transporting that to the final stack of the Golgi apparatus. Now, the Golgi apparatus can also receive these transport vesicles from the smooth endoplasmic reticulum. So I mentioned that with the rough endoplasmic reticulum, it makes proteins. The smooth endoplasmic reticulum is primarily involved in making lipids. And so just like from the rough ER, from the smooth ER, vesicles can pinch off and send their lipids to the Golgi apparatus. And so those lipid products get passed through the Golgi apparatus from one stack to the next, and they get modified along the way. So once again, they might get a carbohydrate added to them to make something called a glycolipid, or they might have something else added to them. So they just get modified in some way. And they're also going to be packaged and prepared to be sent out to another structure. So ultimately, where do these vesicles go? So these vesicles are containing these products. Why do we care? So one purpose is that some of these substances are going to be exported out of the cell through a process known as exocytosis. And so what happens is that a vesicle is going to bud off, and that vesicle is ultimately going to fuse with the cell membrane. So whatever is in that vesicle is ultimately going to be exported out of the cell. If it's something that's actually embedded in the membrane of the transport vesicle, then it might actually get incorporated into the cell membrane. So, for example, this kind of protein right here might actually become a protein associated with the cell membrane. Yet another possibility is that these vesicles that are formed might become another organelle called a lysosome, and this only happens in animal cells. And lysosomes contain digestive enzymes. And these enzymes help to break down either old damaged organelles that need to be broken down and recycled, or they might actually be involved in digesting nutrients that the cell has taken in, or it might be involved in the immune response. So, for example, some eukaryotic cells, such as the white blood cells in the body, can actually engulf foreign particles like a bacterium, and then the lysosomes can break down that bacterium in order to neutralize it. So, we'll talk about that a little bit more on the next slide. Let's take a closer look at lysosomes. So as a reminder, lysosomes are only found in animal cells, so animal eukaryotic cells. So they are essentially bubbles of membrane that contain digestive enzymes. And we haven't talked a lot about enzymes yet, but enzymes are proteins. And so the enzymes actually come from the rough endoplasmic reticulum, because remember, the rough endoplasmic reticulum has those ribosomes which produce proteins. Lysosomes also have a very low pH, which means that they are very acidic, which helps them perform their function. So if we were to draw the membrane of a cell here, and we'll pretend like this cell is something called a phagocyte. And you can learn more about phagocytes when you learn more about the immune system. And so what these phagocytic cells are able to do is they have these lysosomes present. Remember that these lysosomes have these digestive enzymes in them. Now, these cells are able to engulf a foreign particle. So let's say we have a bacterium, and this would be a bacterium that would potentially cause you harm. So what these cells can do, they can pinch in their cell membrane to actually engulf the bacterium. So ultimately what would happen is that a vesicle would bud off, and that vesicle would contain this bacterial cell. Then what could happen is that the lysosome could fuse with this vesicle. And in doing so, these digestive enzymes could kind of take over this bacterial cell and ultimately break it down. So it essentially disables or breaks down or digests this bacterial cell so that it cannot cause harm to the cell. All right, so let's do a quick summary of the endomembrane system. So first of all, it consists of membrane-bound organelles in eukaryotic cells. And these membrane-bound organelles include the nuclear envelope, also called the nuclear membrane, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, transport vesicles, lysosomes in animal cells, and the cell membrane, kind of. So recall that the rough endoplasmic reticulum produces proteins. And the smooth endoplasmic reticulum produces lipids. And in both cases, the proteins and the lipids are going to be either exported out of the cell or incorporated into plasma membranes associated with either the cell membrane or other organelles. The Golgi apparatus modifies the proteins or lipids received from the endoplasmic reticulum. And packages them and sends them out to where they are going to go. So we'll say and packages and exports them. Transport vesicles are just these little bubbles of plasma membrane that help to transport the products from one structure to another. Lysosomes contain digestive enzymes and acid. And their function is to break down pathogens such as bacteria, nutrients that are brought into the cell, or breaking down and recycling old organelles. And then the cell membrane, remember, it just receives a lot of those products. Produced by the endoplasmic reticulum and then sent to the Golgi apparatus. A couple of the kind of functions or benefits of the endomembrane system is that it compartmentalizes chemical reactions occurring in the cell. And in doing so, this increases the efficiency of the cell.